Welcome to Study Loop.
Discussion, we first have to discuss amino acids. So we can break our amino acids down into either polar amino acids or nonpolar amino acids. And these are the main two groups for our amino acids. Okay, polar and nonpolar. What does this mean? Well, polar is going to be hydrophilic, water-loving, and that's just because water is a polar molecule, and uh, like will mix with like if we're talking about miscibility, and in that sense, both will be able to combine in the same environment. Whereas nonpolar amino acids, they are hydrophobic in that they will not be miscible with water. So if we were to look at a protein, for example, our hydrophobic amino acids will be towards the interior of the protein so that it will not have to come into contact with the water environment that surrounds that protein. Now we can further break down our polar amino acids into those that are charged. So that can be positive or negative, and those that are uncharged. And of course, our nonpolar amino acids are all going to be uncharged. So now the question is, how many amino acids do we really have? Well, in nature, we find approximately 20 amino acids. And we find that the breakdown is actually very symmetrical. We find 10 polar amino acids and 10 nonpolar amino acids. Now, of course, we can produce synthetic amino acids as well that are not going to be found in nature, but the big number here to remember is 20. There's 20 amino acids. So let's think about what the tRNA is going to be responsible for doing. Well, our tRNA is sort of like this, and it will be charged with an amino acid. So this is our tRNA, and this is our amino acid. And in order to place this amino acid, in order to charge this tRNA and place the amino acid on the tRNA, we need what is known as a T-aminoacyl transferase. And this is an enzyme. This is a specific enzyme that will allow for this particular amino acid. Let's say it's going to be leucine. Okay, so for example, leucine. And we need to place this leucine on our tRNA. Well, we have a specific aminoacyl transferase that will transfer, just as it is, as you can derive from the name, it will transfer this leucine to the tRNA molecule. And we have, of course, 20 of these enzymes. And that's really what they are. These are enzymes which catalyze the charging of each tRNA with a particular amino acid. So we have 20 of these aminoacyl tRNA synthetases, transferases, and they will correspond to each amino acid individually. So that means for our answer, we find that we will actually have 20 individual amino acids, and that means that there will also be 20 aminoacyl transferases. So that makes our correct answer choice A.